My name is Matt Leevy, and I am the Director of Biological Imaging here at the University of Notre Dame. In the past 10 to 15 years, we've seen a dramatic amount of growth within the preclinical imaging space. We've seen a lot of the clinical modalities become adapted for use on the preclinical side, and those would include PET, SPECT, CT, MRI, ultrasound. But we've also seen a number of modalities created on the preclinical side, and those would include luminescence, fluorescence, perhaps optoacoustics as well. And we make use of all of those modalities on a daily basis to do non-invasive imaging of preclinical specimens. So we can use our PET and SPECT for molecular or functional imaging. We can use MR to look at soft tissue. We can use CT primarily to look at bone, but also to look at soft tissue. And of course, we can do whole animal fluorescence for a range of probe studies or cancer studies, in addition to luminescence as well. And those are what we use on a daily basis here at Notre Dame. Right now, I see a couple of trends that I'm very excited about. Perhaps a subtle but important trend is that the ease of use of all this equipment is dramatically increasing. For example, on my Albira PET SPECT CT, I have faculty, graduate students, postdocs, undergraduates, and even high school students using this equipment independently to execute their own research projects. And the same applies to another system that we have, which is the ICON MRI. Here we have a system, once again, that is now benchtop. It does not require special space. And at the end of the day, the software is easy enough for the broader research community to use. That's a critical factor in continuing to grow the preclinical imaging space as it can permeate further into the research community. The second level of that is that as the tools are getting easier to use, I see multimodality being dramatically enhanced. So over the past few years, we would think of multimodality as maybe luminescence plus fluorescence or PET with CT, having all these platforms adjacent. Now what we're seeing is PET, SPECT, CT, and MR all on one mouse. And it's going to continue to go from there and grow from there. And when you figure, a 10-minute PET scan, a 15-minute SPECT scan, a 10-minute MR, a 5-minute CT, you can get access to all of this information about your disease model in under an hour and do it in a non-invasive fashion that will allow you to examine that specimen over time for the length of a study. That really gets me excited. I think the barriers to utilizing all of this high-end imaging equipment have been dramatically reduced and are continuing to be reduced. More researchers will be able to make use of this equipment, and furthermore, we'll be able to cite more of this equipment at universities that are not primarily affiliated with medical institutions. So we're going to see the imaging community continue to grow, and as it grows, we're going to see an increase in the use of multimodal strategies and a range of experimental models.